It's a full-time paid internship opportunity funded by the DOE. You get to work closely with scientists. So I had a mentor, Dr. Elaine Wong. And I also got to network. I had meetings with certain other mathematicians and scientists, such as Dr. Juan Restrepo, Richard Archibald. So it was a great opportunity to, I guess, get my foot in the door right over I know. It's offered in the fall, spring, or summer. It's full-time. So if you want to do a fall or spring semester, it could be a good idea to take a gap semester. And it's hosted at 17 different national host labs or facilities. So you'd have the opportunity to travel as well if you were to apply. My project was a combinatorial problem. It was focused on enumerating integer partitions and over partitions. It was a pure math project, and it did not have any DOE applications, which is uncommon if you're working at the DOE. But since it was funded by the Department of Energy and not mentor funded, I didn't need specific program funding. So I got paid to do pure math, and it was a lovely opportunity. So I feel like it's useful to define integer partitions. I've provided the definition here, but I feel like it's probably better for our sakes to give the informal definition. So informally, an integer partition is a way to split an integer into a sum of non-increasing integers. So for example, for integers 0 through 5, and we're focused on positive integers. This is a table of all of the possible partitions. So we have, I guess, here, this would be the partition 2 plus 1, or for 5, 2 plus 1 plus 1 plus 1. And so you list them from greatest to least, so you don't have different permutations of the partition. Yeah. And they can also be represented graphically. Again, I provided the definition of a Farrer's graph, but it's not really useful until you look at a Farrer's graph. So this is a Farrer's graph of a integer partition of 18, that specifically being 4 plus 4 plus 3 plus 2 plus 2 plus 2 plus 1. And you can see you stack the boxes vertically down in decreasing order. So 4 plus 4, 3, 2, 2, 2, and 1. So this is all still background. There's, I guess, quite a bit of background for this. But before you learn over Gaussian polynomials, you have to learn Gaussian polynomials. So denoted as the following, Gaussian polynomials enumerate the number of partitions which fit inside an m by n rectangle. So if you go back to the Farrer's graph, this fits into a 7 by 4 rectangle. That's the idea. And it's denoted using this notation. This is read m plus n choose n, just like you would read n choose k, but it's in square brackets instead of round brackets, and it has the subscript q. They're Gaussian polynomials, but they're also called q binomial coefficients interchangeably, because they use the subscript q, and they use q for a variable. This is their formulation. So they're formulated quite similarly to n choose k, but instead of factorial notation, it's in q Herkomer factorial notation. And instead of counting the number of ways to choose objects where order doesn't matter, they produce polynomials such as this polynomial formed by 6 choose 2. Here m is 4 and n is 2. And what this represents here, if you look at this specific term, the exponents are the integer being partitioned, and the coefficients are the number of partitions which fit inside the m by n rectangle. So there are 3 partitions of 4 which fit inside a 4 by 2 rectangle. And so my project focused on over-partitions, the unimodality of, I'll get to unimodality, but Cortiel and Lovejoy define over-partitions as a non-increasing sequence of natural numbers whose sum is an integer, and the first occurrence of a number may be overlined. So the first occurrence of a distinct part of a partition may be overlined for over-partitions. So here we have the partition 1 plus 1 of 2, and then the last occurrence of 1, or equivalently the first, it doesn't really matter, maybe overlined. I think the over-partitions of 3 here are the most interesting, right? So you have this partition 2 plus 1, overline 2 plus 1, 2 plus overline 1, or overline 2 plus overline 1. So when you're doing like regular combinatorics, how many ways are there to choose 0 things out of 2? There's 1. How many ways are there to choose 1 thing out of 2? There's 2. And then 2 out of 2, there's 1 way. So it's combinatorial. But this was my way of representing over partitions using Farrer's graphs. I'm kind of proud of it. But instead of overlining for the Farrer's graph, you put a little star where they're supposed to be. So this is 2 plus 1, overline 2 plus 1, 2 plus overline 1, and then overline them both. And these are these polynomials, which, so this is still read m plus n choose n, but they are over Q binomial coefficients, also called over Gaussian polynomials. Here, overline piece of n of m comma n denotes the number of over partitions of an integer fitting inside an m by n rectangle. So as opposed to partitions, we're thinking about over partitions. And instead of the simple n choose k q Pokemer factorial notation, we have a sum and a series of, it's a rational function, and it generates these over partitions. So another important definition is unimodality. So a sequence of numbers is called unimodal if up to a certain index it shows non-decreasing behavior, and then after that index it shows non-increasing behavior. So the sequence 1, 1, 1 is unimodal, 1, 2, 1 is unimodal, increasing then decreasing, or it could stay the same then decrease, etc. And the unimodality of Gaussian polynomials has been proven by a variety of methods. I believe the most recent method to prove the unimodality of Gaussian polynomials was by my mentor and 2 of her colleagues. They used a computer algebra approach, but it's also been proven via Lie algebras and different abstract mathematics as well. So my goal was to use Dr. Wong's computer algebra approach to prove the unimodality of over Gaussian polynomials. So it was done for Gaussian polynomials. Now we got to do it for the over Gaussian polynomials. And the main feature of that computer algebra method was the cylindrical algebraic decomposition command in Mathematica. What they did for Dr. Wong's proof of the unimodality of Gaussian polynomials was they were able to break up the so you have the sequence of numbers by the coefficients and they enumerate the over partitions. So for example, we have this polynomial right here. They would take half of the polynomial, break it up into a system of consecutive inequalities via the CAD, and then they use that to prove the whole polynomial was unimodal for any m and n. So my goal was to do the same for over Gaussian polynomials. Were we successful? Not yet. But nonetheless, I learned a lot. There were no DOE applications yet, but I don't know, combinatorics is not necessarily only pure math by any means. And so there's potential. But nonetheless, I would strongly encourage anyone who's interested to apply for SULI. I feel like it was a very enriching experience for me. And you can apply for up to 2 years after you graduate. So I got experience to add to my resume. I have a mentor who I still communicate with. She's been very helpful with the process of applying to grad schools. Yeah, and I learned a lot about my favorite thing ever, which is math. So it was really great. I would definitely encourage anyone to apply. I just, Google SULI capital.
Thank you so much, Esther. All right. And so I did forget to mention this, but we're going to hold all of our questions until the very end, so everybody has the full time to speak for their talk. So come on up. So now I'm going to introduce our youngest speaker today. This is Jordan Crain. He is a sophomore biochemistry major, and his research advisor this past summer was Dr. Ann Ray. I should mention that this research was funded by Department of Education funds for improving post-secondary education grant. And what Jordan decided to do with those funds is investigate various protein functions in Arabidopsis thaliana. And without taking up any more of his time, I'll turn it over to him.
Okay, so basically today I'm going to be talking about the research project I worked on over the summer. So let's begin. So first I'm going to talk about the background, and then I'm going to go into the methods and the results and discussion, and lastly I'll talk about the conclusions that I took away from this project. So what was the goal of this project? It was to determine if the protein of interest, what UBC, functions in the peroxisomes of Arabidopsis thaliana, which is the model organism for plants. So what is the role of UBC? So first in this picture, it shows the ubiquitin proteasome system. And so first you have E1s. So those are enzymes that activate a small protein called ubiquitin to get it ready for activation, as you can see here. And then you have E2s, which take the activated ubiquitin and carry it to the E3. And UBC is an E2, which stands for ubiquitin conjugated enzyme. And then the E3 allows the target proteins, as you can see here, to be marked for degradation, and that helps attack the ubiquitin to these target proteins to be ultimately degraded by the proteasome. So where does UBC function? There is some data that suggests that it functions in the peroxisome of plants. So PEX4 is another E2, similar to how UBC is an E2, that is also a peroxin. So that just means that it is a protein functioning inside of the peroxisome. And UBC has a similar homology / sequence to PEX4. So those reasons suggest that UBC potentially functions in the peroxisome. So the schematic basically shows beta oxidation within the peroxisome, and the general function of beta oxidation is to break down fatty acids to produce a form of energy that can be used by the cell in different processes. But also, more specifically, beta oxidation can be used in the peroxisomes of plants to convert IBA to IAA, which are plant hormones. And IAA is the active hormone that IBA converts to through beta oxidation, and that directly controls the root links of plants. So this figure highlights the preliminary experiment that I performed an experimental replicate on through my research project. So you can see here, these are the genotypes they use. So you have wild type, then you have the mutants UBC1 and UBC2, and then you have PEX4. So when you look at the no hormone condition, which is denoted by the black bars, you can see that the mutants are longer than wild type, which is to be expected because studies have shown that the mutants are longer than wild type, but we don't know the mechanism of action why that happens yet. And then when you look at PEX4, it's the shortest because it is deficient in beta oxidation. And then also, when you look at the IBA and IBA conditions shown here, here, and here, you can see that for the wild type and mutant, the IBA and IAA make the root link shorter. And this is because, again, IBA is converting to IAA, which makes the root link shorter. And then when you look at the PEX14 mutant, the IBA bar is the same height as the no hormone bar in terms of root length. And this is because the PEX14 can't convert IBA to IAA, so it's going to stay the same height in terms of root length. But when you do supplement it with just IAA, it will shorten the root length as expected. So these are the overall methods that I did. So first you sterilize and stratify the seeds. Then I prepared the media, plated the seeds, and then prepared and analyzed photos for the data analysis. So to go more in depth with that, the first thing I did was I separated the different genotypes into separate tubes. And then within each tube, you have to surface sterilize the seeds with ethanol and then bleach. And then you perform washes within the tubes. And then after you do that, you stratify the tubes at 4 degrees Celsius. And that just ensures that the germination rates are relatively uniform among the seeds. And then to prepare the media, it was pretty simple. All you have to do is create the agar-based media solution, and then you have to autoclave it to sterilize the media. And then you pour the media onto the plates, and then you allow the media to solidify. And after the media is solidified, you can begin plating the seeds. Then once you do that, the seeds were grown vertically in a growth chamber, which is shown here. And then after that, you can begin periodically assessing germination. And we assess germination every 24 hours in this experiment. And then after that, as you're assessing germinations, you will begin taking photos of the plates. And then I used a program called Fiji to analyze the root links. And the way I did that was I had a ruler off to the side of each plate. And then I told Fiji what 1 centimeter was. And then you can see this red line. So this represents, I went into the program and traced the root myself. And then Fiji will tell me how long that trace was. And that will let me know how long the roots were. So this graph is basically just an overall view of what my results look like, and we'll go more into depth here in a second. So these pictures just represent the different genotypes grown on no hormone, and this just highlights how the mutants root lengths do appear to be longer than the wild type root lengths. And this is the same for the second mutant also, how it appears longer than the wild type's root lengths, and then also how PEX14 has the shortest root length, which was also supported by the preliminary data. And then, so when you're looking at no hormone, you can see that the UBC1 and 2 mutants are longer than the wild type, and this is to be expected and is supported with their preliminary data. And also how PEX14 is the shortest in terms of root length. And this is also because it is deficient in its ability to perform beta oxidation. So now when you're comparing the IBA and IAA conditions among wild type, the root lengths are shortened compared to no hormone, which is also supported by the data because beta oxidation causes IBA to be converted to IAA, which shortens the root lengths also. And again, this is supported for the mutants and also, or for both mutants, UBC1 and UBC2. And then PEX14 can't convert IBA to IAA, so it doesn't shorten the root length, which is why these are relatively the same length. And then when it is exposed to IAA, it is already exposed to IAA, so it will inhibit the root length. And then now we're going to compare the ratios between no hormone and IBA, like we did with the preliminary data. So if you pay attention to this ratio here between no hormone and IBA for wild type, so you can see how this ratio for mutant 1 for no hormone and IBA appears to be greater than the ratio between wild type. And this is also consistent with the second mutant. So because of this, it ultimately suggests that the mutants perform beta oxidation better than wild type. So that suggests that the original function of the UBC gene is to somewhat inhibit beta oxidation. So overall, I contributed to the preliminary evidence of this research project, and my experiment added to the potential conclusion that this protein may be functioning in the peroxisomes of Arabidopsis thaliana. However, more experimental replicates will need to be performed to determine if the ratios are statistically significant, which I'm currently pursuing now. And then I would like to acknowledge Dr. Ray for helping me with the research process and giving me the opportunity to partake in this research project. Dr. Shu for collecting the preliminary data for this experiment while working in Dr. Hughes' lab at Michigan State University. Dr. Siopsis and Dr. Gibson for applying to the grant for this research, and then the FIPSE grant for providing funding for this research project over the summer. And yeah, that's all.
Okay. So,
My present.
go to, all right, awesome. So it's also my pleasure to introduce Mackenzie Nicholas. She is a senior biology major and has also been a huge part of the all things MC band the past 4 years. Her research advisor was Dr. Jennifer Brigati, and her research was also funded by the Department of Education FIPSE grant this past summer. She spent her summer extending her senior research project, which involved testing for the presence of Naegleria fowleri in Northeastern Tennessee waters.
Thank you. So good afternoon, everybody. I have spent the past almost a year testing for the presence of Naegleria fowleri in Northeastern Tennessee recreational water sources. The first half of the year was just researching. The summer was mostly going out in the field, collecting samples, and then this semester was optimizing and using that protocol. So what is N. fowleri? Well, it is the brain-eating amoeba. You can see the brain-eating amoeba right there. The top picture is in its condensed state. The second picture has a little protrusion, which is called pseudopodia, meaning false feet. That's how it moves throughout its environment. N. fowleri is classified as a free-living amoeba, meaning it's living in its environment, moving around with those pseudopodia. N. fowleri is thermophilic, meaning it's like warm water, specifically fresh water. The main thing that N. fowleri does is it causes primary amoebic meningioencephalitis, which is shortened to PAM. In order to be infective, it has to enter deep into the nasal cavities. So using a neti pot, jumping into a lake where the water gets shot up into your nose. PAM is the reason for this study. And the reason why it's so important is because PAM is extremely fatal. It's around 97% fatal of all the cases. But however, it is really rare, with around 200 cases worldwide since 1965, around 100 of them were in the US. Another important reason to study Naegleria fowleri is because global warming occurs, and it could increase the number of cases of PAM in the next couple of years because the water warms, therefore Naegleria fowleri likes warmer water. There's no treatment. They have tried antifungals like amphotericin B, antiparasitic drugs, antibacterial drugs, but there's no real treatment for PAM. So the CDC recommends that you use a nose plug when swimming in warm, fresh water, or just don't jump into those bodies of water. Basically, if we find N. fowleri in a body of water, we can notate it so that the public knows to take extra precautions when using that water. I just want to say, brain-eating amoeba seems really, really scary, but it is super rare, so it's not something that you guys should be worried about. Spoiler, I did not find it, so that's good news. Also, this is a BSL-2, biosafety level 2 organism, so I had to make sure I wore proper PPE when I was experimenting with it, like a gown, gloves, working in a negative pressure biosafety cabinet, wearing goggles, and sometimes even a face mask. There's been no lab-acquired infection, so everything was perfectly safe. These were my methods. I gathered 500 mils of water from each source. I went to 20 different places. I went to 5 different sections of each site, gathering 100 mils from each site and then combining them through a cheesecloth to get rid of excess big debris. Then we used a filtration process to concentrate those free-living amoebas. And it was filtered through a 0.22 micron nitrocellulose filter, so it would have trapped everything bigger than a virus on top of that filter. We then take all the stuff off the filter and put it on a non-nutrient agar plate with E. coli, and then let it sit in the incubator and observe it daily for 2 weeks. And eventually we would start to see some feeding trails, which you will see a picture of coming up soon. That was where the amoeba had eaten a line through the E. coli, and you can see their little paths traveling through the plate. It's really cool. So once we found those prospective positive plates, we flooded those plates with sterile water and extracted the DNA from those plates, which then we were able to do a polymerase chain reaction to amplify that DNA. PCR, polymerase chain reaction, is basically taking a tiny piece of DNA and making it really big so that you can see it. We targeted general free-living amoebas, Naegleria genus, and N. fowleri specific. And then to visualize that DNA, we ran it on HL. These are all of the sites that I visited this summer. This is a picture of Augusta Quarry. I thought that was the most beautiful one. It's a place that I have swum before. As you can see, we took a lot of the samples in July, August, in September a little bit. And we made sure that the water was around 80 degrees Fahrenheit because that is the preferred temperature for Naegleria fowleri. 12 out of the 20 sites that we sampled had feeding trails. These are the feeding trails. I'm not sure how well you can see it on this screen, but there should be like little tiny lines all across the plate. Can you see the lines? No? Okay, well, they're there and they're cool. Sorry, you can't see them. So all of these had the cool little lines. They were all at the 37. So once we were able to extract that DNA and perform the PCR, we ran it on a gel. This is a perfect example of one of the gels because it has the ladder positive and negative controls as well as the experimental results. So in the first two wells is a negative and positive and Fowleri specific control. Wells 4 and 5 were the positive and negative general FLA controls. And the last three wells were the experimental controls from the Greenbelt Lake, which is, if anyone's been to Vienna, it's right by the library, that lake right there. That is what's in that lake. There was no Fowleri specific, no Naegleria, but there were general FLAs. This is for the results for all 12 of the sites that had feeding trails, which we extracted DNA from. So we did not find M. Fowleri. None of the samples were positive, but we did find 4 of the samples were positive for Naegleria genus, meaning it's a close relative to M. Fowleri. Those are not infective to people. We haven't found another species of Naegleria that is infective, but it shows that, it is a potential habitat for M. Fowleri. And 9 of the 12 samples were positive for general free-living amoeba. And here is a graph that separates the PCR results based on temperature. There's sample size of 6 in each temperature group. As you can see, there's more presence of FLAs in the warmer temperatures than in the colder temperatures. For 6 versus 3. Still no Naegleria fowleri in either one. So over the summer, I completed 145 PCRs, 31 gels, and I looked at 50 plates daily, did 27 DNA extractions, and I spent over 250 hours in lab. Each PCR took, if I ran on average 10 PCRs a session, they were about 3 hours each. So it's a lot of time, a lot of effort. These are just some of the many gels that I ran. But I had an absolute blast. So Naegleria, in conclusion, Naegleria escimi and FLAs were found in the local waters, but not the infective brain-eating amoeba. However, it may still be present in those waters. I only took 500 mils from an entire lake or an entire river, so I could have just not gotten it. Also, I could have just not extracted enough DNA and it wasn't showing up in the amplification. So it's always a good idea to be precautious, just in case M. fowleri is present, even though it is rare. It's just a good idea to stay cautious of that. Specifically from my summer experience, I saw gorgeous places in Tennessee that I never even knew existed, like the Covert Concord, Legal Point Park, and it was just an incredible experience. I have always been a self-identified lab girly, where I don't like to go outside, and I thought that I would be in the lab all day and I wouldn't do anything outside, but I learned that microbiology isn't always just in the lab. I went outside a lot. We walked a lot of miles, encountered a lot of confounding variables like big storms and trees blocking our paths. This is when we were going to a quarry and we just couldn't sample from there because there's a large tree in the way, couldn't drive there. And I learned, most importantly, that I really want to work in a lab for the rest of my life because I just had an incredible experience and I loved every second of it. I just want to say special thanks to Dr. Brigati. She's not here right now, but she was my thesis advisor. She gave me this idea for this project, helped me all the way. And thanks to Dr. Ray for helping my PCR troubleshooting and being there through all the tears and all the happy laughs. Hannah Phillips, my lab partner, who was there with me all summer, really helped with everything that I did. The FIPSE grant for the summer funding and the senior study grant for funding for supplies. Thank you guys. Are there any questions? Oh, I guess we're wait till the end quick.
Mackenzie brings up a good point. If our other speakers want to join her up there, we have plenty of time from these perfectly timed presentations to ask you all questions. Caitlin?
I want to know how you chose your locations. Did you specifically go for not flowing water, or did you kind of combine and do some of each?
This is all of the locations. I basically chose whether people interacted with the water. Like at Greenbelt Lake, Alcoa Duck Pond, that's a really big fishing area. Whereas Fort Patrick Henry Lake, Boone Lake, that's a big swimming area. I picked areas that were known for recreational use. So that, if I were to find Naegleria fowler, I could put a warning like, hey, be careful. raining amoeba, like. So,
Which places were positive for the Naegleria genus amoeba? There was 4 places.
yes. They're there. The Covent Concord, Louisville Point Park, Fort Patrick Henry Lake, and Bays Mountain Reservoir. Those last 2 are in Kingsport. Those first 2 are in Maryville. Apparently, I've never swam there, but apparently Louisville Point Park is like a big swimming boating area. So, yeah.
This is for Esther. I am not the most mathematically inclined. I can get some things there. Thank you for that. I actually understood it, so that was awesome. Did your project, did you love math? Did it make you want to continue math, or
Oh, yeah.
was there anything that you learned that you didn't like versus what you did like?
Well, I enjoyed it. I mean, there were certain like logistical things about like being in the lab that were, I don't know, like I was in a little cubicle. I mean, it was my cubicle, but it was still a cubicle. So I don't know, if they let me do some stuff from home too, because I didn't need to use their facilities necessarily. Like it was like completely sufficient for me to use my like hand-me-down laptop from my sister. So but all of the math I really enjoyed.
Awesome.
Thank
How
you.
did it change your perception of careers in math? Like actually seeing like a laboratory full of mathematicians going to work?
Yeah, that was really great to see. Actually, I knew for my whole life that I wanted to do math, so I was like, okay, I'm gonna be a high school math teacher. Do I want to do that? By no means. Like I don't want to do that at all. And but my mentor spends all day doing math and computer science, and I think that's beautiful. And like, there's not just one way to do it too, which is great to see.
Well, Esther was talking, I have a little bit of it's not Dr. Friend. I understood that, so there's a result for these non-overlined partitions.
Yeah.
And then you're trying to get the same result for the overlined partition. So why the overlined partition? So why is that an important thing to look at, or is it just an interesting thing to look at that's just kind of for fun?
It's math for its own sake, in my experience. Like I said, there was no DOE applications, but it's kind of like confounding in a way, because unimodality proofs are kind of like notoriously difficult. So I don't know.
I guess what I'm asking is not so much for an application, because it's pretty clear right now we don't have one.
Yeah.
That doesn't mean there will never be one. I'm more asking, is there a different mathematical context under which this kind of new arrangement matters?
Yeah.
And if you don't know, that's okay.
I don't know.
Okay.
I have a question for Jordan. Is there a difference in the number of seedlings that you get for the various mutants, or for?
We tried to keep it around the same number just to keep it controlled. Like we aim for around 60.
Okay.
Sebastian, so you were saying they were waiting on getting more people to replicate what you had done in the lab.
Yeah.
What does he do as what would happen if they found?
We would just do different experiments to also further prove if it functions in peroxisomes, but maybe not through just a different overall experimental design than what I did, to just prove if it functions within the peroxisomes.
John?
Does it function anywhere else in the plant, or is it just like there's
Yeah,
this thing and we
there have been some screenings that show that it also can function in the nucleus, and there are some experiments right now to see if it does function in the nucleus, but what I was doing was just to look at if it functions in the peroxisomes.
All of John?
Not really a follow-up, just a different question. What was the plant that you used? You said something about it being like the base plant, or I don't
Oh,
really
we used
remember.
Arabidopsis thaliana. So that's just a model organism that's used for plants.
Why is it a model organism?
Um, that's a good question. I'll have to get back to you on that.
I can answer. That's my plan. It's really small, so you can grow a lot in a, small or big area. Has a really fast generation time. The genome is entirely sequenced, so there's all these, mutant collections and tDNA insertion collections, so we know what all the genes are, we just don't necessarily know what the functions are. So fast generation time, fully sequenced, small, grows quickly, easy quote to take care
The Drosophila plants.
of. Exactly.
It's also for you.
Yeah.
I can see you doing this so early in your college
That's
career.
thanks.
That's crazy. Would you recommend other people to do the same?
I would say yes, because it really helped me get out of my comfort zone of just like getting that research experience early on so I can decide what I want to do in my future. So yeah, I would definitely recommend it.
What did you need to change about your schedule to fit research in?
I need to change about my schedule.
And I could go for all of you, like I knew you did this over the summer as well, but a lot of you are continuing this on and or extending projects, so you're still working with your mentor. So how do you fit it into your schedule now? How does it?
I had to put some classes off that I wanted to take earlier on to make time for the research. So yeah, I guess that's how I mainly would change my schedule around.
Hopes and prayers. It's a lot of work, especially for an entirely lab-based senior study where you have to optimize the I created the protocol, I optimized the protocol, and then I did it. So it's a big time commitment. But thankfully I was paid for it over the summer for the FIPSE grant.
So for me, I applied to a few other internships at ORNL because it's like local. But I didn't get either of them, so my schedule did not have to change at all. But my mentor is just kind enough to stay in communication and answer questions and stuff. So that's been really nice.
I think from my observation of what Jordan was doing, he really took a lot of his weekends or holiday times or summer, he carved out a lot of time that other people might be resting. Poor Jordan. But he's right now taking so many difficult classes at the same time, and he still was able to figure out, okay, where is this piece going to work? What's the timeline? When you sit down to do an experiment, you have to think about how long does it take to do X? By this date, am I going to be here? Right, Mackenzie?
Yeah. I was doing homework in the lab, my PCR was running, I was doing homework.
Right, it's hard, and then you can't predict things, and things don't work the way you expect, right, Jordan?
Yeah, not always. What other questions do we have? We have some final question. Well, let's give our speakers a round.